I'll give you a little history on Dyadic. As the slide shows there, we do everything from stonewash genes to genes used for biofuels, feed, food, all kinds of applications. I'm happy to say that I was here at the inaugural Bio conference 13, 14 years ago. And the testament is we're still here. So for all you guys starting up and beginning your businesses, there is hope. We have a lab in Wageningen in the Netherlands. So for the Europeans here, we do lots of collaborations with institutes, researchers, and companies. You'll see that later. Our headquarters in Jupiter, Florida. For all the Canadians and the Northerners, come wintertime, come down and visit us. We've got you covered both ways. This is just a slide kind of hitting on what everybody's talked about. We're in the middle of a genomic revolution, and we're finding genes every day from nature that can do all kinds of wonderful things. It's exploding at an accelerated rate. It's going faster than Moore's Law did for the computer industry. And if there wasn't enough natural diversity, which they're talking about today, from volcano vents down to the deep sea, etc., when you do protein engineering and you shuffle and screen all these proteins, you're making exponential numbers of variants that can do the things that carbonic solutions are doing right now and everything else. But you'll see coming up here, this exploding GNA pool needs to be done and created, and you have to produce these things. And the bottleneck really has been, and they kind of led up to this, if you can't produce or express proteins in large enough volumes and low enough cost, you're stuck in research. And the vision that I had over 20 years ago was to compete with the giants, the Novozymes and the DuPonts of the world, is to develop a world-class protein production system. And we set out to do that first and foremost in the blue gene business because we were making enzymes to stonewash genes. And it turned out to be, and this is the one slide takeaway that everybody in this room should imprint in their mind, is that we've been able to achieve levels of expression of 100 grams per liter in 6 or 7 days, where 80% of that protein is one protein, one enzyme. That is a remarkable number. If that's not good enough, we've been able to produce that at 500,000 liters, and we've been running at 150,000 liters for almost 2 decades. So this is a world-class expression system that allows the industry access to compete with the big guys, because it's a $500 million to $1 billion investments that have been made by the DuPonts and the Novozymes and the DSMs to play ball. And most people in this room can't afford a fraction of that. We couldn't, certainly, when we started out, and we got lucky along the way in some of these cases, but the message here is that we can work together with you to bring your discoveries, whether it's a natural gene, an evolved gene. We just need to know what gene you want to have produced and for what reason, and figure out a way to share in the value of that. And if we can do that, we can all create value for not only ourselves, but for the world as well. The other thing that you need to do, which we've already done in this system, is we not only made it robust, productive, scalable, programmable, it's safe. We've gone through GRAS, US FDA, with an enzyme to prove the safety and effectiveness of the enzyme being produced. And there's only a handful of organisms in the world that have actually had that stamp of approval, and we have that. So we have the world-class production system. It has the safety pedigree that you need. We just need the genes and the ideas and the thoughts from all of you to figure out what to program it with to create new products for the world. So this slide is basically a slide about biofuels and about how getting off oil allows us to not fight wars in Iraq, not spend our money on putting soldiers on the ground, not have all this economic turmoil that we all grow into. We can create renewable sugars, first generation from corn and wheat and starch, second generation from cellulose. And my vision, as it was 20 years ago, next vision is that we're going to engineer crops and plants so that they almost digest in water easily. And you can get cellulosic sugars to produce 85% of whatever is needed that's being made today by oil. And we're playing a part in that in the digestion of the sugars from the plants with our enzymes. And of course, this is one of the topics, greenhouse gases are harming the planet. It's harming the people that live on the planet, the way we breathe. If anybody's ever been to Beijing 10 years ago, let alone today, you can't breathe. This is a major problem. It's a growing problem. It's degrading the icebergs, the polar bears, everything you can see here, the crops. the agricultural growth, dry climates, genetic engineering of crops you'll see coming in. And here's an example. This is here today. Abago, which is one of our licensees, producing enzymes at 500,000 liter scale to digest wheat straw, sugarcane bagasse, corn stover, switchgrass, etc., any lignocellulose material. You can reduce up to 86% of the greenhouse gas emissions with renewable fuels made right in your backyard, whether it's America, Australia, Brazil, or Europe. We can grow our own fuels, which means we're not dependent on oil from foreign countries. We're stimulating economic stimulus, providing jobs and growth in the rural economies. It's just a phenomenal benefit for society, for humankind, and for all of us. And I think it's here today. It's getting better, quicker, faster, and cheaper. And we're proud to play a role in that with Abago and others. It's not just about biofuels. Enzymes in this space, we also have enzymes that work for municipal solid waste and in biogas. And the Europeans among us know that there's about 3,000 biogas plants in Germany. Our enzymes work better, faster, and cheaper because they are developed in a neutral and alkaline environment where the enzymes from trichoderma will not operate. So from a biogas perspective, in breaking down lignocellulosic biomass, whether it's MSW or biogas applications, C1 enzymes from Dyadic are the future, and they're here today. So this is sort of a controversial slide outside of this room. Inside this room, everybody believes that I think GMOs, whether it be crop genetics or fungal genetics or bacterial genetics, are here to stay, and it's actually a safer way to make things because we know exactly the changes we made versus randomly making those changes and not having any idea, which is how the world operated for the last several hundred years. Scientifically, we all think this is a better way. We all know that this improves the quality of being able to make more nutritious, healthier foods, but also the ability to feed the world with a growing population. Enzymes play a role in this. Either they can be cloned into a plant, with companies like Agravita are doing. They can be produced in C1, which is what we're doing, and obviously the Novos and the Genicors and other people also make microbial enzymes for feed. They digest grains in a way that allow them to become more nutritious and allow you to get better weight gain for less. So we can feed more people, we can do it more nutritious, we can do it healthier, and this is again growing quicker, faster, and cheaper, and we're able to do the things we couldn't do before, whether it's GMO or non-GMO. Here at Dyadic, we also make non-GMO enzymes, so for the people that do not want GMO, we still do make non-GMO enzymes. We make them, sell them all over the world in 35 countries. We have distributors and formulators that implement those into their products, but we also make GMO-type products because we do believe that that is a better way to do it for the future. Biosimilars, not a big topic here at this bioconference, but of course at the bioconference in Philadelphia. It's all about making drugs. And we're going to talk about why our technology is really, it's all about, we don't care if the gene's a human gene, a fungal gene, a gene out of a volcano, or where it comes from. If there's a use and a benefit to society and mankind, we've developed a way to mass produce those things into proteins and enzymes at massive scale. So we believe, and the study shows that over the next decade or two, that there's $250 billion worth of savings that you can get by making biosimilars. And obviously we believe that using our technology can do it faster, quicker, and cheaper. And to give you an example, Chinese hamster ovaries are how they actually make a lot of these protein therapeutic drugs today. We believe that C1 can make them at 10 times larger scale, at 1/10 the cost, with 1/10 the media cost, without having the prions and purification scheme. And the genetic engineering and synthetic biology and all the genomic revolution, we can program this cell line to make drugs quicker, better, faster, and cheaper. And we can also do that for vaccines. We're working with Sanofi Pasteur, one of the largest companies in vaccines, to develop a new vaccine and a new system to make it quicker, faster, and cheaper, because obviously if we have, sorry, I gotta go backwards, epidemics that hit us, like Ebola that might have come, we're not prepared. Because the systems that exist today can produce small quantities, treat a small amount of people. Our technology can do it at massive scale, can be implemented in just about any fermentation system in the world. There's idle fermenters sitting everywhere, and we can actually address the problems that we may all face as a society, and I think we need to do it sooner or later. And I think here at Dyadic, we're working towards that. So this is kind of an interesting slide, because we cannot solve our problems with the same thinking we used when we created them. And I think this is the problem we all have. I'm a journalism major that wrestled at the University of Iowa. I'm not a biochemist. I had no background. I have no reason to really be here, but I am. Just as science is evolving, we as humans are evolving. And what we know here is we know we don't know everything. In fact, I took the approach that we know nothing. And we went out and sought the people in the world that actually knew things that we needed to know. And we took advantage of those relationships, and we leveraged those relationships. Whether this slide shows into cellulosic sugar with companies like Abengoa and CIMB, the world's leaders in this space, or industrial enzymes with companies like BASF, one of the largest chemical companies on earth, or in the vaccine space with Sanofi Pasteur, or anybody sitting in this audience or in a university, anywhere, or industry lab across the world. They're discovering incredible things, and they're all coming up with the same problem. They can't make sufficient quantities of proteins. That's the vision we had 20 years ago. We can do it at a large scale, we can do it affordably, we work with the best in class, and here's an example of 10 EU-funded programs that we have going on now that include other commercial partners like Procter & Gamble, universities like Wageningen University, etc., doing all kinds of wonderful things, creating all kinds of products. So we can do that with people in this room, other universities, other institutions, other industrial partners. It's your imagination is really the only thing holding you back. And we worked with some of the best and the brightest minds to put this together. Remember, we took the approach that we knew nothing, and we went out and found people that knew something that we didn't know and made it better. Trust me, we now know a lot, but we still work with collaborations and with institutes and the scientists and the scientific community to achieve all the wonderful things that need to be done. And Bio has been a great platform for that. The Bio Partnering, if you haven't been involved in that, is a phenomenal platform to introduce yourself and what you can do to companies like ours and others. And I thank you.